Hello everyone, today we're going to talk about DNA replication and we're also going to talk about DNA mutations. Our DNA is a double helix, meaning that our DNA is made up of two strands and those two strands start to twist around each other. Helix meaning that those two strands are twisted around each other like you see right here. Our DNA is made up of a monomer, or you can use the word building block, known as nucleotides. What is a nucleotide? A nucleotide is made up of three parts. A phosphate group, which is made out of a phosphorus element bound to a bunch of oxygens, a sugar group, and a base. So a phosphate group, a sugar group, and a base, those three parts right there connected to each other is considered one nucleotide. Now, does your DNA contain only one nucleotide? Of course not. It's made up of several of these nucleotides. That's why we say poly. Poly meaning many. So if you look at the DNA strand right here, we say that these strands right here make up the backbone of your DNA. So we have two backbones right here. Those backbones of your DNA, they're made up of sugar groups and phosphate groups just repeated. So you have a phosphate bound to a sugar, bound to another sugar, bound to another phosphate, etc. So right here, those repetitive sugar and phosphate groups make up the backbone of our DNA. Now, right here, sticking out of the backbone of your DNA, you have the bases. DNA has four bases: adenine, cytosine, thymine, and guanine. So you have those four bases sticking to the backbone of our DNA. Now, let's go ahead and take a look at this image, which is just a bit more zoomed in. So you see the double-stranded DNA right here. You can see the backbone of the DNA, which is made up of repetitive sugar and phosphate groups. And then sticking to the backbone of the DNA, we have the bases. As we said, our DNA is double-stranded. So you can see one strand right here, and you can see the other strand right there. Both strands are going to have the backbone of phosphates and sugars. So you can see right here, here's the backbone of one strand, and here is the backbone of the other strand. And sticking out of the backbones, you have your bases. As we said, DNA has four bases: adenine, guanine, cytosine, and thymine. Now those bases, if you take a look, they are actually connected to each other via a bunch of hydrogen bonds. So hydrogen bonds are going to hold those bases together, creating a base pair. Now adenine will only bind to thymine. Guanine will only bind to cytosine. So if one strand of your DNA reads AGTC, your other DNA strand is going to read TCAG, because T-thymine is going to bind to adenine, cytosine binds to guanine. If you look at both strands, you will notice that they are not identical. They are complementary to one another. Identical would mean that if this strand of your DNA reads A, then the other strand of your DNA would have to read A. If that's the case, then we would say that both strands of our DNA are identical, but that's not the case. Adenine will bind to thymine and guanine will bind to cytosine. So we say that our DNA strands are complementary to one another. And again, those bases are going to be held by a bunch of hydrogen bonds. Easy way to remember which bases will bind to each other. You can remember the mnemonic apples on the trees. So A will always bind to T, and you park your car in the garage. So C always binds with G. Apples on trees, car in the garage. Okay, so let's go ahead and build a complementary strand together. So as we said, our DNA is double-stranded. Those two strands are not identical to each other, they are complementary. So if your DNA reads the following, ATTCCG, what would the complementary strand be? So adenine will always bind to thymine. Whenever you have thymines, you will have adenine on the other side. Whenever you have cytosines, you will have guanines. And then, of course, that G will bind to a C. So this right here is the complementary strand for your DNA. So your DNA would look something like this with two strands that are complementary to each other. Before a cell splits, the cell has DNA inside of it. And so before the cell split, it would have to duplicate that DNA within it. As we said, the DNA is a double-stranded helix. The very first step to start DNA replication is to go ahead and split that DNA helix. You want to split the two DNA strands. The enzyme that would actually help split the two strands for you is an enzyme known as DNA helicase. After the two strands are split, another enzyme will come and help build the new complementary strand, and that enzyme is known as DNA polymerase. So polymerase comes in and start helping build that new strand for you. So you can see right here, here is the old DNA strand. Let's say the old DNA strand is depicted by the color blue. So we have those two old blue DNA strands right there. And we want to go ahead and we want to duplicate those two DNA strands. The very first step, as we said, is to go ahead and split those two old DNA strands apart. And the enzyme that would help with that is DNA helicase. Then, after you split those two old DNA strands, to each old DNA strand, you want to go ahead and you want to start building a new complementary strand. The enzyme that would help build the new complementary strand is DNA polymerase. So at the end of DNA replication, you would end up with an old DNA strand bound to a new DNA strand. And you will have an old DNA strand and a new one on both sides because, as we said, we duplicated both DNA strands. If your old DNA strand reads an A, then your new strand will carry a T. If your old DNA strands reads a G, then your new strand is going to read a C, as you can see right here, because the new strand that you are building is complementary and not identical to the old DNA strand. Now, to make a protein, we go through a process, two processes actually, transcription and translation. Transcription happens inside of the nucleus and translation happens in the cytoplasm. Transcription is the process of taking your DNA and making a messenger RNA out of it. After you build that messenger RNA, the messenger RNA leaves the nucleus and it will go through a process known as translation to make a protein. In our body, we have so many different proteins, And so whenever you want to make a protein, you have to go through the process of transcription and translation. To build a protein strand, we have to go through the process of transcription and translation. Transcription is where we're going to build a messenger RNA strand from our DNA strand. So we know that our DNA strand consists of 4 bases: adenine, guanine, cytosine, and thymine. RNA also consists of 4 bases: adenine, guanine, cytosine, and uracil. So RNA does not have thymine, instead it has uracil. When you build a messenger RNA from your DNA strand, let's say that your DNA strand reads the following: A, T, C, G. To build the messenger RNA, adenine on your DNA will bind to uracil. Thymine will bind to adenine, cytosine will bind to guanine, guanine will bind to cytosine. So your messenger RNA strand will read UAGC. Messenger RNA does not have any thymines. So adenine will always bind to uracil if we're trying to build a messenger RNA. And you have an example right here of a DNA strand and the messenger RNA strand that will result after going through the process of transcription. Now, to read the messenger RNA that was just built, the messenger RNA is read 3 bases at a time. Those 3 bases are going to be known as a codon. So 3 bases at a time, we refer to them as codons. Each codon is going to code for an amino acid. When you put a bunch of amino acids, you link them together. This is how we're going to build our protein. So proteins are made out of amino acids, and we get those amino acids by translating the codons of our messenger RNA. Each codon codes for a specific amino acid. To figure out which amino acid is going to result, we have to look at this chart right here, known as the codon chart. So let's say that your messenger RNA codon is UCA. What you want to do is look at this chart and first of all, try to figure out how to read it. So right here on this side is going to be the first base of the codon. Up here is going to be the second base, and over here on the side we have the third base. So let's go ahead and try one together. So if my codon reads UCA, that means that my first letter is a U, or my first base is a U. So you would want to find the first base. So here is the first base right there, the U. Your second base is a C. So here is C right there. So after you find the U and the C, you would kind of like connect them together, and that means we are in this block right here. And then all you have to do is go ahead and find exactly UCA, which is right here within this block, and it would code for an amino acid known as serine. Let's try another one. Let's say that your codon is ACA. So your first letter or your first base is A. So you would find the first base right here, A. The second base is C. So C is over here. That means that I am within this block right there. So ACA, you would go ahead and highlight ACA, which is right here. And it means that ACA codes for amino acid known as THR or threonine. Let's try another one. Let's say that your messenger RNA codon is GUG. So your first base is G. So here it is, G. So you try and find it. So here it is, G. Your second base is a U. So U is over here. And so you would go ahead, can I connect them? That means that you are in this block right there. So GUG, which is right here. Here is GUG, which means that it codes for the amino acid valine. High exposure to UV rays or X-rays or certain chemicals can lead to a genetic mutation. A genetic mutation is basically any change taking place in our DNA sequence. A substitution type of mutation is where one base or more than one base gets substituted for another base. An addition type of mutation is where an extra base or more than one goes ahead and gets added to your original DNA sequence. Deletion is where one base or more than one gets deleted out of your original DNA sequence. To build our hemoglobin protein, which is the protein found in our red blood cells, that protein is going to help us transport oxygen. To build it, we have to read the DNA, build a messenger RNA, and then start building our amino acid chain. Now, if someone goes through a mutation and their DNA sequence of their hemoglobin gene goes ahead and gets mutated, and you can see that it's a substitution type of mutation. That change in the DNA sequence is going to result in a change within the messenger RNA sequence, which is going to result in a change in the amino acid chain. When someone goes through this type of mutation right there, this could result in a type of disorder known as sickle cell anemia, where those individuals are going to have a hard time transporting oxygen. This image right here shows you the effect of mutations on our amino acid chain, or basically on our proteins. So if the top image right here is showing you the original messenger RNA and the original amino acid chain that would result, the images below it is showing you the effect of the mutations on the amino acid chain. So as we said, when we read our messenger RNA, we read it 3 letters at a time, our codons. So if we look at this codon right here, originally it was supposed to read GGC. If someone goes through a mutation, a substitution type of mutation, and that G got substituted for an A, you can see that the amino acid became different. From glycine, it got switched to serine. Now, if we go ahead and actually delete a base, that's going to result in a more disastrous effect. Why? Because if you delete a base, you still have to read your messenger RNA by threes. And so therefore, if we go ahead and now divide our messenger RNA in 3, notice how originally we were supposed to read triple U. Now, instead, because you deleted that U, you're going to have to move to the next codon. And so you're going to have to take a letter from that next codon. So now your codon is going to read UUG. But not only that, you've messed up all the codons that would come after that. Notice how this last codon now only has 2 letters or 2 bases, because you keep taking a base from the other codons to make up for the one that you lost. Same thing, more disastrous effects will result if you also insert an extra base. So if you insert an extra base, again, you're not just going to be affecting that one codon right there, you're going to be affecting all the other codons that are going to be after the insertion of that base. So as you can see, both the insertion and deletion can lead to a more disastrous effect on your amino acid chain compared to the substitution type of mutation. If you take a look at the codon chart, what you will notice is you can actually have several codons coding for the same amino acid. If we take a look at the amino acid proline, notice how there are 4 codons that code for the amino acid proline. Why is that a good thing? Just in case you go through a mutation, which is going to result in changing your codon, you might be lucky, and the codon that would result is still going to code for the same amino acid. To give you an example, substitution mutations could have different effects, a silent, a missense, or a nonsense type of effect. Let's go ahead and look at the silent effect or the silent mutation. So if this is the original DNA, and after building the messenger RNA and looking at the chart, the amino acid that resulted was arginine, and we go through a mutation, and that mutation resulted in a change in our messenger RNA after changing that base right here within our DNA. The messenger RNA that was built was AGA. If you look, you would find that AGA is also going to result in the same amino acids, meaning that this is a silent type of mutation because your amino acid never got changed. So this is actually pretty good because if your amino acid never changes, then your protein is going to stay exactly the same. So it's actually very beneficial to have more than one codon coding for the same amino acid. As you can see right here, just in case a mutation takes place, we could still be lucky enough to get the same type of the amino acid. If our original DNA sequence coded eventually for that arginine amino acid right there, and we went ahead and we went through a mutation where this time the mutation resulted in a different amino acid, because let's take a look at the chart right here. Originally your messenger RNA was AGG right here, which codes for arginine, and we went ahead and we went through a mutation where now our codon reads AGC. Notice how AGC does not code for arginine, it codes for serine. That means that this mutation resulted in a change in your amino acid sequence. And so when this happens, we call this a missense type of mutation. Let's say we go through a mutation and that mutation results in a codon. And when you look at this codon right here in the chart, it codes for stop. What does that mean? Stop. Let's say that originally your amino acid chain is made out of 4 amino acids. And you go through a mutation right about there, and you only get 2 amino acids, and then it results in a stop. That means that you cannot proceed any further. And that means that instead of having a chain of 4 amino acids, now you only have a chain of 2 amino acids. Definitely, that's going to be a pretty short sequence compared to your original amino acid sequence, and that's definitely going to change your protein. So when you get a stop type of mutation, we call it a nonsense mutation.